the block on metabolism with an introduction to digestion and some energetics and molecules you're going to need to be familiar with throughout all of metabolism. So the first thing to ask is, what is digestion? In the simplest terms, you're taking complex molecules and tearing them apart, doing some chemistry, breaking bonds, making new bonds, possibly doing oxidation or reduction reactions, and you're turning them into smaller components of themselves. Right? We like to say in class, we'll say break down all the time. Right? The molecule doesn't really fall apart. We're doing chemistry to it, but colloquially we will say break down. What we mean is large complex molecules become smaller, simpler molecules. And for the most part, the one we're going to concern ourselves with is when we turn those larger complex things into the molecule acetyl-CoA. So a 2-carbon unit, the acetate group, attached to a carrier molecule called coenzyme A. Right? You don't have to memorize the structure of CoA, but know that it is a carrier molecule that carries around carbon structures for me, such as the 2-carbon unit, the acetyl group. Okay? And then the last thing we'll talk about for this exam is we'll take the acetyl-CoA units and oxidize the acetate group completely. Those 2 carbons get oxidized completely to CO2, and we produce some electrons from that. When we oxidize a molecule, we extract electrons. The extraction of those electrons gets sent down a chain, and ultimately we give the electrons to an electron sink. And in our example here, we're going to use molecular oxygen, which when you add electrons to something, you're reducing it. So we're going to reduce molecular oxygen to water molecules. And the movement of those electrons down a chain pumps some protons across the membrane, will allow that proton difference across the membrane to power a machine, which makes ATP. Right? It assembles an ADP and a phosphate. So we're going to turn some chemical energy into a gradient, into mechanical energy, turning a machine, physical turning around, into chemical energy again in the form of ATP, and it's very efficient. And so to start with, we need to take some gross molecules, gross in the sense that you can see them, they're not microscopic. So some large molecules, such as food, and start tearing them apart into smaller pieces. So the picture on the bottom right is what some people would consider the world's perfect food. It was in college, right? So it contains a little bit of everything. You've got your carbohydrates, you've got some protein in there, you've got nucleic acids because those are cells, and it's got lipids all over it as well. So it's got a little bit of everything you need, possibly not in the right proportions, but all the things are there. And you need to start giving your enzymes access to these molecules. So the first thing you want to do is completely denature everything. Okay? But in order to do that, we need to get it homogenized. Right, so what that means is we need to turn it, basically blend it. So if you could turn your pizza into and throw it in a blender and make a more liquid pizza out of it, doesn't sound very appealing, but who knows, someone will come up with that idea and it'll be popular. But if you could turn it into a more of a liquid, homogeneous, blended mess, then you'd have an easier time digesting it. And that's mainly what you're doing when you're chewing food, right? You're blending it, making it more homogeneous, right? You're also adding some enzymes. You're also changing the pH when it gets to the stomach, and then when we get past the stomach, we'll change the pH again, right? So, and add more enzymes. But we need to start homogenizing the food so we can have access to it by our enzymes in order to start digestion. Okay? So let's say our food enters the stomach, and it's a very acidic environment, right? So it's mostly hydrochloric acid and around pH 1 to 2. And so most proteins at that pH will denature, right? They'll completely unfold. Their primary structures, you remember, are still intact. So we need to add some enzymes to start digesting those peptide bonds. And those are our proteases. We've talked about chymotrypsin already and its mechanism. But chymotrypsin isn't born active. It starts out life as a zymogen. A zymogen is a precursor enzyme. So, and the example on the bottom, when chymotrypsin is made as a protein, it's actually made as chymotrypsinogen, and then a piece of it gets cleaved off by yet another protease, and it becomes an active chymotrypsin. And all of these are like that. They come in a precursor form, right, inactive form, and a piece gets cleaved off by yet another protease, right, and then it becomes an active form like pepsin or trypsin or elastase and so forth. Right? So the reason for this is we don't want them active enzymes the moment they're made inside the cell, or they would just destroy the cell. So they're made as a inactive, too long version, right? A piece of the protein needs to be cleaved off for this to work. And so these are made in the pancreas and stored there, and along with some other things stored like bicarbonates and things from the gallbladder we'll talk about later to help lipid digestion. And they're excreted through the bile duct into the beginning of the small intestine, or the duodenum. And the purpose of this is to start the digestion of these peptide bonds that survived the pepsin in the stomach. Because remember, these enzymes are very specific about what they cut. They don't cut any peptide bond. It matters what amino acids surround it. It has to fit in that specificity pocket. So here we have additional enzymes that can take up the slack where pepsin and perhaps elastase or trypsin can't cleave this protein. Chymotrypsin and carboxypeptidase can. And the combination of all these enzymes chews up your protein into much smaller pieces, maybe a di and tripeptides at that point. You do get some individual amino acids as well, but your protein of say 300, 400 amino acids is not completely intact anymore. You've unfolded it due to the acidity, and all these enzymes have chopped it up into manageable pieces, maybe single amino acids, dipeptides, tripeptides, pentapeptides, right? So small pieces, nothing longer than a few dozen pieces anymore, a few dozen amino acids. Okay. So here's another chart showing that at the top, the cascade of events that happens. So we release this enzyme called enteropeptidase, and it's starting the process. It turns trypsinogen, that's the zymogen, into an active trypsin. Trypsin can also activate more trypsinogens. So once you get one started, they can activate all the rest of them. And then trypsin, like any good protease, cuts all the other things it has access to. So it turns proelastase into an active elastase, carboxypeptidase into procarboxypeptidase into carboxypeptidase, and so forth. So all of these things turn on other enzymes. And why have this elaborate cascade of events? Again, we want amplification. I want one trypsin becoming active to turn on all the others in that released bile. Okay? So the food enters the small intestine. We suddenly have a acidic environment entering the small intestine. That's dangerous because you could destroy the lining of the intestine. So we stimulate the release of bicarbonate. The bicarbonate will neutralize the hydrochloric acid, right? And we'll get sodium chloride and carbonic acid, which is not so bad, right? It's just CO2 and water. And then we'll neutralize the pH to a more manageable range, somewhere around 6 to 8, rather than pH 1 or 2 like it was. So we don't degrade our own small intestinal lining. Okay. How is this accomplished? Well, when the cells that line the beginning of the duodenum detect the change in pH, because they can measure it from the solvent, they release cholecystokinin. Right? Cholecystokinin is a hormone, right? So it's released by these intestinal cells into the blood, and it goes to the pancreas and says, hey, we need you to release bicarbonate and zymogen so we can digest this food, this chyme that's being sent our way, right? And it uses the mechanism we talked about before in the last lecture, where it binds a receptor, sends a signal inside the cell, and so forth, right? I won't repeat all that for you. But ultimately, these enzymes degrade the proteins into either individual amino acids or oligopeptides, which is just a fancy way of saying a small number of amino acids hooked together, not a large number, 1, 2, 3, 4, so forth, right? And then on the surface of the intestinal cells, also another peptidase, right? Another protease that'll degrade these oligopeptides into di and tri or individual amino acids. Well, those things we have transporters for, because they normally can't cross the membrane. We learned that. So we get some transporters to import these amino acids into the small intestinal cells. Inside, we have more peptidases that degrade all these tri and dipeptides into individual amino acids. So all the proteins you've eaten are now just a flood of amino acids. What those intestinal cells do is they keep a little tax for themselves, whatever amino acids they might need, and they send the rest on the backside of the intestinal cell into the bloodstream. Well, all the blood that leaves from there, from the backside of the intestinal cells that line the entire intestine and stomach, has to go through customs first, because this just entered the body. The lumen of the intestine is in fact outside the body, right? It's the same thing as your mouth, right? Inside your mouth is not a cavity of your body. It's open to the environment. So the entire digestive tract is actually open to the outside environment. There's no cell protecting it. So anything that goes through these intestinal cells must get screened. Now, not so much amino acids, they're pretty safe, but any other chemicals or drugs that you're taking, right? Or any kind of molecule that goes through the ingestion pathway has to go through screening. So it sends it to the blood here, and that goes to the liver. So all those blood vessels coalesce into the hepatic portal vein, which goes right to the liver, and the liver acts as a customs or an immigration station. And so it screens all these molecules. Now, if it's amino acids and sugars and things like that, those are normal, it lets them go. But it filters out a lot of the drugs you take orally. That's why an oral dose of a drug is generally much larger than, say, an IV dose, because you've bypassed the liver's customs by giving an IV. Okay? So what about carbohydrates? So we talked about protein, we break it up into amino acids, but what about carbs you eat, right? What the crust of that pizza? So it's full of things like starches and glucose and maltose and other complex sugars, but we'll focus on just glucose for now, or combinations of glucose like starch. And starch is just a long polymer. We'll talk about it later when we get to it, but it's just a long polymer of glucose. Right? It's connected mostly by alpha 1,4 bonds, and then you get an occasional alpha 1,6 bond to make the branch points. Okay? If you eat these things, your pH won't break these bonds. We need a certain enzyme that'll do this. So in the intestine, we release an enzyme that's called alpha amylase, right? And it can break most of the bonds of starch. There are a few it can't get to. For instance, it can't break that alpha 1,6 bond, and it can't break an alpha 1,4 bond if it's not surrounded by other sugars. So in this example, these green arrows is where it could cut, right? But if you get a maltodextrose product, the alpha amylase sees it as too small to fit in its active site. It would try to cut these bonds, but it's just not long enough. If it had 4, it might cut it in half. But 3, it just doesn't cut it very well. Again, you have limit dextran here in the middle. It can't break the 1,6 bond, and the 1,4s are too close to the end to cut. And if you end up with a maltose, of course, it can't cut that either. So alpha amylase needs some assistance in degrading this starch down to only glucose. So we have a trio of other enzymes that help it out. We have dextranase, which comes along and breaks this alpha 1,6 bond, leaving a pair of maltose molecules. Right? The maltose then can be broken down by maltase to give you glucose. And then the alpha glucosidase can come along and cut off one of the 3 of maltotriose, again leaving maltose for maltase to handle. Now, alpha amylase does generate some glucose occasionally by cutting it off, and we'll talk about those later. But you have an ability with these set of 4 enzymes to turn any complex carbohydrate made of glucose into just a series of glucose molecules. And so we can handle amino acids and glucose in our metabolism. So far we've broken down 2 of our food groups. Right? The third type of food we're going to have are lipids. Right? So we had the carbohydrates, we had amino acids from proteins, now we're going to have lipids. We're going to ignore nucleic acids because you really don't run out of those, and you can make your own. It's not a big deal. So we're not going to talk about metabolism of nucleic acids so much as far as food, but the other 3 categories we will. Right, lipids are unique among these because they don't make polymers. However, they're not water soluble. So we need to get our lipid in an emulsion, right? So it's like Italian salad dressing, right? If you let it sit long enough, it'll make 2 layers, and the only place that the enzymes will have access, let's say the enzymes are water soluble, the only place they have access to your lipids is at the interface between the 2. So if you had a cylinder of Italian dressing and you let it settle, and there were enzymes in the water part that digested lipids, they can only access the lipids at the interface. So it's a very small area of interface. We'd like to speed up that reaction by giving more of our enzymes access at any one time, so we make an emulsion. In simple terms, we stir it up, right? So the small intestine doesn't have a stir or a fan in it. You don't shake it around too much. You could go jogging, that might help a little bit, but you might cause other problems like all the food coming up. So we add a molecule that does the emulsion instead. So these are emulsifying agents, like the one you see at the top there. That looks like the word chocolate, and it's not. It's glycocolate, but it will help you digest the fats in chocolate. So if you had to guess, what is it made from? Looking at the structure alone, what is that molecule synthesized from?
Cholesterol.
Cholesterol?
From cholesterol, exactly. It's that steroid core. You even notice on number 3, we've got our OH that's always been there. You could number these rings if you had to, right? And then it's still got its tail down here. But we've made some modifications. This OH down here and the one up top were not there before. Those have been added to our cholesterol molecule. Our tail has been altered as well. We've added something to the end of the tail. What is this thing I've added to the end? What's this group on the end of the tail? The nitrogen and the 2 carbons.
Carboxylate ion.
Say again?
The carboxylate ion.
No, you've seen it before. It's a little bigger than just a carboxylate. It's a nitrogen.
Oh, the amide?
Well, it's a nitrogen on an alpha carbon, a carbonyl on an alpha carbon, and 2 hydrogens on this carbon. Does that help you see it? It's the reason we call it glycocolate.
Is that glycine?
That is glycine, exactly. So glyco sometimes means sugar. In fact, most of the time means we added a sugar to something. But it could also mean glycine, right? So here's our case of glycocolate. Colate is the conjugate base of cholic acid, which is a cholesterol acid. So our tail has been turned into a carboxylic acid initially. And then we decarboxylate it, make this amide with glycine to make a glycocolate, right? And this is one of your many bile salts that made in the liver, stored in that gallbladder, and released whenever you eat fatty foods, right? That cholecystokinin will cause it to release. Right? The other enzymes there are going to be lipases, right? So the glycocolate is giving you more access, vastly increasing the surface area between the water-soluble and the lipid-soluble stuff, because parts of it look very water-soluble and parts of it are not. So it bridges the gap between the two, making an effective emulsion. If you happen to have your gallbladder removed, right, you don't make as much of this, so you can't store as much of this, and so you can't go out and eat a whole cheesecake, which I don't recommend if you have your gallbladder. But you don't have the ability to digest fats as efficiently. So if you eat a very large meal full of fats and you don't have a gallbladder anymore, you can't effectively emulsify the food, and as it makes its way through your body, it won't be digested and it just exits the other end as undigested fats, which is not fun. So if you know someone who's had their gallbladder removed, think about where you might want to take them to dinner first. They generally want to eat small, many meals instead of big, 2 or 3 meals a day. Okay, so my mom had this surgery a few years ago, and that's why I'm mentioning this. So at the bottom, we have our lipases. We can take our triacylglycerol, which is another of our lipids, and cut off all those fatty acids one at a time until we end up with a diacylglycerol, monoacylglycerol, and then just glycerol. Right? So what did we do with those? So here's our triacylglycerides at the bottom. We add water and all those lipases. We turn it into individual fatty acids, and then our monoacylglycerols, or just glycerol. Those are imported into the cell because we have transporters for those things. And then this might sound unusual, but once those are inside the cell, they're reassembled as triacylglycerols again. We just took them apart in order to put them right back together. The point of that is to get it across the membrane, because we couldn't drag it across the membrane as a triacylglyceride. Okay, so we break it up into its constituent parts, the fatty acids and the glycerol. We can import those, and then we reassemble it. On the back side of that cell again, we take these triacylglycerols. They aren't really water soluble, right? So in order to get them to the liver, we need to give them some kind of taxi or carrier. And that's where our chylomicrons come in. We'll talk about these later as well, but they're just carriers, and they take these triacylglycerols to the liver. Okay? Same in the bloodstream, but the triacylglycerols needed a carrier because they weren't water soluble. Okay? The other enzyme we'll have will be phospholipases. These are digestive enzymes that are found in the venom of snakes, bees, wasps, scorpions, things that inject their prey with digestive enzymes. So if you think of it in one way, instead of just eating something, they inject their digestive enzymes into the organism before they eat it, and then they'll swallow it later, right? Or in the case of, let's say, a spider, they inject it, it completely digests the organism first, and then they just drink it, right? So they're effectively doing the digestion outside their body, at least the start of it. So in the venom of a lot of these organisms contain phospholipases, and you notice there's A1, A2, C, and D here. There's no B because A1 and A2, one was named B and originally found they were working on the same one, so they just called them A1 and A2. It's the quirks of naming things in science again. But you see they cut an individual ester or phosphoester bond along the molecule. So this is your typical two fatty acids stuck on a glycerol with a phosphate and then some alcohol X on the other side. So this is a typical membrane lipid. And two particular cell types that these phospholipases go after, right? They lyse red blood cell membranes and they lyse nerve cell membranes. So if your prey is, say the snake here bites a little rat and the rat starts to run away, well, the rat is clearly faster than the snake, right? But if you digest the rat's nervous system and red blood cells, the muscles aren't going to work very well anymore. They won't be told what to do, and they won't be supplied with oxygen, and the snake can catch up with its prey. Right? They also have other things in the venom besides the phospholipases. We have all sorts of digestive enzymes in there that break down bone, connective tissue, collagen, everything. Right? So they swallow the organism whole, and it just digests slowly. Right? They don't emulsify it like we do. Okay. Here's that cholecystokinin pathway on the right. I'm not going to read it all to you, but I want you to recognize it, that it's binding to a 7-transmembrane receptor. We activate that G protein. The G protein eventually activates protein kinase A, which turns on phospholipase C. And if you remember the slide brought to you by the letter C, all of that takes place. And we ultimately turn on protein kinase C. Protein kinase C tells these little zymogen granules that are in the cells to move to the surface and release your contents. So effectively, you told the cell, by releasing cholecystokinin, you told other cells to release the digestive enzymes that they're holding in little vessels. And it's all done by this amplification pathway we talked about last time. Right, so let's move on to metabolism. That was just digestion, an overview of how we start breaking down molecules. But metabolism is now what we do with those small molecules, right, such as glucose or acetyl-CoA. Right, so what do we do with these molecules? We can do two things. We can take the molecules and break them down further, do more oxidation in general, or do more degradation into smaller pieces, and that's called catabolism or catabolic metabolism. So think catabolic means generally oxidizing things or making things into smaller pieces, right? And your goal is to extract energy from it or extract electrons from it, right? It's our reducing power. Anabolism is just the opposite, or anabolic metabolism is just the opposite. Here we take building blocks, smaller molecules, and assemble them into larger molecules. Generally, we use reduction reactions to do that. So it's kind of the opposite of catabolism. An easy way to keep these straight is if you watch sports at all, you've probably heard of anabolic steroids, right? These are, molecules and drugs that athletes take against the rules to enhance their performance. And a lot of them build muscle. So if you think anabolic is the case of building something, whereas catabolic is breaking it down. Okay, so shown on the right is the pathway we'll do next time. We'll take glucose, and across 10 steps, we'll turn it into pyruvate. This is going to be glycolysis. And then pyruvate could have several fates, as 2 of them are shown here. We can do aerobic metabolism and turn pyruvate into acetyl-CoA, right? Or we could turn pyruvate into lactate. And that's anaerobic or fermentation. And we'll talk about those next time. Okay, so what is metabolism? Is it something that everybody can control mentally? No. Is it something that can go up or go down, or some people have a higher metabolism than others? Not really. What we really mean by it is all the processes that go on in the cell, all the chemistry that takes place inside a cell in an organism. It's not something you have conscious control over. Right? And if you take any organism from an elephant to a bacterium, they all do the same basic metabolism, right? But in E. coli, it seemed like it would be simpler than it would be in an elephant, but it's really not. Of the basic reactions that all cells must do to survive, they're all the same, right? They all must do that glycolysis pathway. They all must make energy somehow by oxidizing molecules or putting it across a membrane or using a food source. Sometimes the food source, in the case of plants, is photons, right? So that provides the energy input. Whereas some people use molecules of energy. Those are non-photosynthetic organisms. So all of these things are used in the same way no matter what kind of cell it is. So it's rather simple in that there are many recurring motifs. There's only so many molecules, only about 100 molecules that we're going to talk about. That might seem like a lot, but it's really not that many. And between those 100 molecules, there's only about 1,000 reactions that can take place. Again, that might seem like a lot, but as far as the cell is concerned, that's not very many reactions. And then we have specialization, right? So this is not on this slide, but we have specialization. So when you get to multicellular organisms, some cells become completely dependent on other cells because they just don't do that job anymore, right? So muscle cells are completely dependent on being delivered their oxygen because they're in an environment in which they cannot extract that much oxygen from their local environment of water or air or whatever's around them, right? Usually not air, right? So they're dependent on other cells to help them out, right? And so here's the same process again, this big chart on the right, and we will cover most of this chart, not all of it, but every dot on this chart represents a molecule, and every line represents some reaction that is taking place. And I know they're not labeled, but there are many charts like this. There's a big poster like this you can look at, download, buy, whatever they try to sell you. But it has all the molecules listed on it and all the reactions. And if you want to get one of those, download those big posters and look at the region that we're interested in. It could help tie this together for you. But the important thing is you notice none of these are in isolation. There isn't a single point up here that isn't connected in one way or another to another system. Right? It's not isolated in one system. So our carbohydrate metabolism, as you see right down the center in the dark, heavy blue line, the first thing we're going to do, that's glycolysis right down the center, is connected to everything else because it's supplying our raw materials for nearly everything. Okay? So most of these are going to be catabolic pathways if they're degrading something, or we're going to call them anabolic pathways if we're building something. Okay? And some of these pathways are sort of hard to categorize. We call them amphibolic. It means part of it looks like a catabolic pathway, but we're actually building something. So it seems odd. There's some oddballs in the list that don't quite fit into our nice dichotomy of catabolic and anabolic. And we'll look at a couple of those as well. Okay. So onto the reactions themselves, we have some energetics to talk about. So it's very simple math. We talked about these before. If in order for a reaction to proceed, remember when we talked about enzymes, the overall delta G needs to be negative. Okay, so an example down here is reaction A. If you have molecule A and some reaction takes place and it's converted to molecules B and C, perhaps it's breaking the molecule into two pieces, the overall delta G of that is plus 5, it says, plus 5 kilocalories per mole. This reaction is not favorable under those conditions. Now we could dump in a lot of A or continually remove B and or C and possibly drive it forward. That's Le Chatelier's principle. But the overall delta G is plus 5. I'd like a better way to do it. So if I take molecule B and involve it in a new reaction, somehow convert B to D, and the conversion of B to D gives you a negative 8 kilocalories per mole delta G. It's very favorable. And that's a coupled reaction I can do because the favorability of reaction 2 is more so than reaction 1 is unfavorable. Right? So if I take the delta G's and just add them together, plus 5 plus a negative 8 gives me negative 3. So if I couple these reactions, I get an overall favorable reaction. So converting A into B plus C, and then immediately have an enzyme there that converts B to D, I've effectively turned B into an intermediate between the two enzymes. Overall, A gets turned into C and D. And it's favorable. So I'm using a thermodynamically favorable reaction, reaction 2 here, to drive an unfavorable reaction, reaction 1. So we couple these together. Right? They're still separate enzymes, but the enzymes are in the same area or compartment of the cell, if we're talking about a eukaryotic cell, and the substrates are shuttled from one to the other very effectively. Okay? The most common one we're going to do with this is hydrolysis of ATP. Right? ATP is not some ball of energy like they like to say in some classes, but it's a molecule, right? It's this molecule at the bottom. It's adenosine with 3 phosphates on it, right? So those 3 phosphates could be cleaved off individually or in groups. So if we take the ATP and cleave off the last phosphate, the gamma phosphate, we're left with ADP and a free phosphate. And that's a favorable reaction, right? You see the delta G for that is negative 7.3 kilocalories per mole. So I could couple this. Let's pretend this is reaction 2, or reaction 2 from this slide. Right? I can couple this hydrolysis of ATP to power any reaction up to a positive delta G of 7.3. But if the reaction requires say a positive 9 to do it, if reaction 1, if we look back here, if reaction 1 perhaps was plus 9 kilocalories per mole, coupling it to a hydrolysis of ATP would not suffice because it's only negative 7.3. I'm still got a positive overall delta G. So if I need to power something that's say plus 9 or plus 10, I can't use 1 ATP in that manner to do it. But I could cut off 2 phosphates at once. So in this case, I take the beta and gamma phosphates and remove them at the same time, right? Leaving a pyrophosphate as a product and leaving AMP behind. But that has a delta G of negative 10.9. That could power something if it's coupled to a reaction, as long as the positive delta G it's coupled to is not greater than 10.9. Okay? If it is greater than that, we may need a new energy source, maybe a couple of ATPs, or 3 or 4 of them. Maybe do it in sequence, make the reaction steps smaller. But I can power something by breaking these bonds. Now, why is breaking the bonds of ATP or GTP or UTP or CTP, why are the breaking of these bonds favorable? Well, if you look, these have negative charges, right? And they don't want to be next to each other. And we also have this 2 phosphoester bonds right next to each other. Again, not very favorable. And we'll talk about more of these in a second. So here's my ATP molecule again shown at the top with the phosphates drawn out and adenosine on the right. And to stabilize these bonds, because it's not a very stable arrangement, I could add small molecules like divalent cations, magnesium for instance. It's not the only one, but it's one of the better ones. So the magnesium here bridges 2 of the anions on the phosphates and stabilizes that arrangement. Now there's a couple places it could bind. 2 of them are shown. It could bind between the alpha and beta phosphates, shown in the top one. It could bind between the beta and gamma phosphates. It could also bridge the 2 gamma negative oxygens. Right? So there's a third place it could bind. It's not shown in the pictures here. Right? So if the ATP can break in multiple ways, how do we know which one the enzyme is going to use? Will it make ADP and a phosphate, or will it make AMP by cutting off the latter 2 phosphates? How does the enzyme know which one to use? Well, that's mainly determined by how the ATP binds in the enzyme's active site. Right? It has to bind ATP in either case, but it mainly positions this magnesium differently. So if the magnesium, which is held by the enzyme, that's its cofactor, is positioned between the alpha and beta phosphates like this, most often the latter 2 are going to leave. Right? And when the beta and gamma phosphates leave, then the magnesium could bridge the 2 oxygens that are left in the remaining AMP. Right? When the magnesium is positioned more between the beta and gamma phosphates, like in the panel here in the center, then it's more likely just the last phosphate's going to leave. And then the magnesium can jump over to bridging the alpha and beta, or the 2 beta oxygens. Right? So depending how the magnesium is positioned, in the enzyme, when the ATP binds, it chooses to use one or the other type, and it generally does only one of them. Okay, so how about the phosphate that leaves? So if we cut off a phosphate, we said this is a very favorable reaction. Well, what makes it so favorable? Why is it so efficient? The phosphate that leaves is now very free. We've given it many more degrees of freedom than it was when it was attached to the other phosphates. Number one, the charges are now farther away from each other. Okay, so these charges on the phosphate that leave are farther away from the ones that remain. They're not next to each other anymore. So that's favorable. Taking like charges and moving them farther apart is a favorable thing to do. Okay, so electrostatic repulsion is great. The other thing is the phosphate that leaves itself has 5 total bonds. The phosphorus atom can make 5 bonds, and it's a tetrahedral arrangement, so one of them is a double bond, right? And remember, phosphorus is not in the same row as carbon and oxygen and nitrogen. It's in the next row of the periodic table, so it has some higher order geometry it can adopt. It can have more than 4 bonds. So it has 5 bonds, one being a double, and there's 4 ways to arrange that, since there's 4 oxygens there. One of them is less favorable than the other 4. The one at the bottom right is not the best arrangement, where we have more separation of charges, but the other 3 are equivalent. And because all these resonance structures exist, right, you remember back from Gen Chem and organic, the more resonance structures you can draw, the more stabilized the molecule is. So we have many resonance structures for this phosphate. I know these might look like the same structure, but it's a different oxygen harboring that extra pair of electrons. Finally, we have hydration. Right? So when this molecule leaves, more water molecules are involved in solvating these things. Right? There's so many water molecules around these 3 phosphates when we start, and then when we separate this phosphate from the others, more water molecules than before are interacting with all these negative oxygens and carbonyls and phosphoonyls. So we have all these water molecules that are preferred to be reacting with these things or interacting with these things. That's a favored change in entropy. Right? So the water molecules would love to interact with these things. The phosphate negative charge has moved away from the other negative charges, and it's resonance stabilized. All these add up to our negative 7.3 kilocalories per mole, or in the case of the pyrophosphate, negative 10.9 kilocalories per mole. Okay. So how does this compare to other molecules? So at the chart at the bottom, I've thrown some other molecules in the list with ATP hydrolysis. So all of these molecules on the table have a phosphate that they could lose, like ATP does. ATP, in fact, has 3, but we're going to talk about losing the last one. So ATP becoming ADP. We said it was negative 7.3. I'm going to use the kilocal on the right. You can use the kilojoules if you want, but I'll compare it on the right. Negative 7.3 kilocalories per mole. All the molecules above it in the chart are more energetic or less stable. They have a higher tendency to get rid of their phosphates. Right? Those 3 would rather get rid of their phosphate more so than ATP does. Right? And the 4 below it have less tendencies, lower energies of getting rid of their phosphates. So what this means is, if any of the molecules above ATP here were combined with an ADP, they could transfer their phosphate, given the appropriate enzyme, to ADP. Because turning ADP into ATP would have a kilocal per mole of plus 7.3. Right? You just do these processes in reverse, you get the opposite sign of the delta G. Adding the enzyme does not change that. Remember, enzymes do not change the delta G. So creatine phosphate, for instance, and ADP, in the presence of the appropriate enzyme, could transfer the phosphate from creatine phosphate to ADP, and my products would be creatine without a phosphate and ATP. Right? Similarly, ATP could transfer its phosphate to glucose. Right? It could be on number 1 or number 6, but it could transfer to it because the delta G of putting a phosphate on glucose, let's say number 6, is a plus 3.3. Whereas the delta G of the ATP losing its phosphate is more negative than that was positive. It's negative 7.3. So I could use ATP to phosphorylate a glucose, but I could not use ATP to phosphorylate a creatine because it takes more energy than it could provide to put it on there. by removing one phosphate. So those three molecules above have more energy than ATP. They're energy-rich compounds. They're great ways to store energy, and we're going to talk about those three in a second. The ones underneath are effectively the products of ATP donating its phosphate to glucose or glycerol or another phosphate to make those molecules at the bottom. Right? So those are the targets of a kinase that uses ATP, whereas the ones above are the substrates, the source of phosphates to make an ATP. Right? So the three above it, I've drawn their structures on the right here. And can anyone tell me what these three structures have in common? And before you give me the obvious answers, which I know you're right, yes, they all have a phosphate. I agree. Right? That's not the question. Based on their structures, what makes them so high energy or unstable? What do you think? Think back to organic. Why are these three so unstable?
Would it be the isolated double bond between the so like looking at phosphoenol pyruvate, the alkene with the two hydrogens coming off of it, with the isolated bond from the phosphate double bond?
What do you mean by isolated bond?
Like sharing two sigma bonds between them?
Yeah, what do you call
Like
it?
you said, beta keto?
Exactly, that's a beta keto arrangement. Exactly, and if you remember, all of these have that. It just they vary on the element that's the double bonded to. In the top case, it's carbon. In the second case, it's nitrogen. In the bottom case, it's a carbonyl. But in all three cases, this is a beta keto arrangement. You're absolutely right. So in beta ketos, as you remember, are not stable. They can break one of the two single bonds bridging the two double bonds. Okay? Alpha ketos, however, were very stable. Those were conjugated double bonds, right? So these are not alpha keto, they're beta keto arrangements, and beta keto is very unstable, thus high energy. And the most likely way to resolve it in these cases is to lose the phosphate group, right? To dephosphorylate these molecules. And so keep that in mind because we're going to use that many, many times to explain things later. We're going to create beta keto arrangements in our chemistry steps with the intent to use that energy that we just put into it in a subsequent step. Okay. So how about making ATP from creatine phosphate? Let's go through that one. Okay, so you saw on the previous slide, creatine has a phosphate attached to it. It's not a very stable beta keto arrangement. And I can take an ADP and accept that phosphate because the delta G overall would be negative. So if you're running some kind of event like the cyclist on the right or a marathon or swimming or whatever it might be, in the first few seconds of what you're doing, you rapidly run out of ATP, right? Like you're running a race, a sprint, and you're going to rapidly run out of ATP within seconds. We're talking maybe 10 to 20 seconds, you are out of ATP, probably within 10, right? So how do you keep going? Well, you have all this creatine phosphate in the muscles. So the creatine phosphate has enough stored phosphate to immediately replenish a lot of those ADP molecules with their phosphate to make yet another round of ATPs available. And that gives you about double the time in seconds. So now you can last 20 seconds instead of 10. This is why sprinting, you can run 10 seconds, not a problem. But if you try to go 20, you're stretching it, right? So sprinting times over 20 seconds for humans anyway tend to drop off drastically after that, right? So if you, to make the math easy, if you run the 100 meters in 10 seconds, you can run the 200 meters in around 20 seconds, right? It's slightly less than that, but it doesn't drop off dramatically. But if you go to 400 meters, that doesn't equal 40 seconds at all, right? It drops off significantly. You're going to go much longer times. And the reason for that is you've run out of creatine. So your muscles need another source of ATP. So the next source is fermentation. We'll do anaerobic metabolism. We'll talk about this in the next lecture. But that's the green line here. So this is fermentation in the muscles, turning pyruvate into lactate or lactic acid. And the muscles are doing that as a stopgap because we need to move now. We need to run away from this bear that's chasing us or whatever it is. Right? If you don't get away, you're going to become food. So we need that anaerobic or fermentation to fill in the gap. If we have to go longer than several minutes, or maybe dozens of minutes, up to hours, then anaerobic metabolism will not suffice because we can't harbor that much lactic acid. We can't send it to the liver fast enough and it's going to cause muscle degradation, right? You're also going to have a huge change in the pH of the blood with all the CO2 there, right? So what we do is aerobic metabolism instead, right? So here we're going to use oxygen as a source and get much more energy out of these molecules than just fermenting it. But that's slow. That's not as rapid as, say, creatine phosphate or the anaerobic metabolism pathway. So it's slow. So if you're running a marathon, you're not sprinting the whole time. You have a consistent, slower pace, right? But if you're in shape for the marathon runners, then that's the pace you run the entire time, right? And you only save that creatine phosphate for the last sprint at the end, right? Okay, so thinking about ATP and how it's used, we said we use it for running or stuff like that, but you use it all the time, right? So if you're just sitting there right now listening to this lecture, at any one time you have about 100 grams of ATP in your body, right? So the average 70 kilogram person, right, has about 100 grams or 0.1 kilograms of ATP in your body, and it's fleeting. It only lasts a few seconds. Right, so we must be constantly making more, which we are. So we use it almost as fast as we can make it, just sitting around doing nothing. Right, so in a 24-hour period, the average person uses about 40 kilograms of ATP. All right, that's 400 times that 100 grams. So in a 24-hour period, in 1 day period, just sitting around, listening to this lecture, sleeping, doing nothing, you rotate your ATP, your turnover of ATP, each molecule was used about 400 times, right? So there's a lot of turnover. And that amount, 40 kilograms, is more than half that 70 kilo body weight, right? So you're turning it over quite a bit. But that's not fast usage. If you're exercising, you consume about half a kilogram every minute, right? Remember, we used 40 kilograms in a whole day. You just used half a kilogram in 1 minute. So if you exercise consistently for an hour, right, you're going to use 30 kilograms just in that 1 hour of what you would use 40 for a whole day. So exercise consumes a lot of ATP. Okay? So where do we get the energy to make more? Okay? So we need to oxidize other molecules, right? And the oxidation of molecules provides some electrons that we're going to use on an electron transport chain and ultimately a proton gradient to make more ATP. But to get those electrons, I need to oxidize something. So what are my foods I could oxidize? Well, we have glucose, we have fatty acids. Let's try to oxidize those things. If you think about a glucose molecule, and you guys think back to carbohydrates, your average glucose molecule, right? The average carbon in a glucose looks like what? What are the things on that carbon? I know there's 6 of them, but on average, what does every carbon look like? If you had to put it on the scale at the bottom, where is glucose on that scale? The average carbon, what does it look like? Which one of those 5 answers?
Methanol?
Methanol.
Exactly, looks like methanol, right? 5 out of the 6 carbons look like methanol stage, right? It looks like it's got one OH on it. One of them looks like the formaldehyde stage because it's an aldehyde, right? But the other 5 look like methanol. So it's 5/6 of the way methanol and 1/6 of it is formaldehyde. So mostly if you had to draw a line, it's just to the right of the methanol on this scale, right? Just ever slightly to the right. So there's a lot of potential to oxidize that carbon. That carbon right now has just one OH on it. We can do 1, 2, 3 oxidation steps to it to get it to CO2. So there's a potential energy of 168 kilocalories per mole there. Right? That's the energy difference in one carbon. And you have 5 of those like that in glucose and 1 in the formaldehyde stage. So glucose has a lot of potential oxidation energy in it. What about something like a fatty acid, right? Let's say our typical 16 carbon fatty acid, right? Where does it fall on the scale? The typical carbon.
Methane?
Yeah, it's methane, right? So of the 16 carbons in a 16O fatty acid, 15 of them look like methane. right? There's no oxygens on the carbons at all. And one of them looks like the formic acid stage, right? The number one carbon of the fatty acid, because it's a fatty acid. But then you have a lot of these methanes that have the high potential to be oxidized 1, 2, 3, 4 times, right, to get all the way to CO2. So the fats store a lot of potential for oxidation energy again. Okay, so we'll use these, and the byproducts of this are going to be CO2, right here in the end, and water, because when we oxidize all these stages of carbon, those hydrogens have to go along with the electrons that are leaving. And we give those electrons ultimately to O2 to make water, right? And one of the most missed questions I always get when I give this talk is the O2 that we breathe in, right? When you breathe in O2, about 99% of it gets converted to what? Water, not CO2, right? 99% of the O2 you breathe in gets converted to water. It gets reduced to water. That's its goal. That's its point in the electron transport chain. It is not converted to CO2. But if you ask that of, say, elementary students and high school students and a lot of college students, they'll assume it's the O2 you breathe in picks up a carbon and it releases as CO2, which is not true at all. Right? The carbon in CO2 comes from the food source, right? And the O2 in the carbon generally comes from water, right? So as we oxidize these things, we add O's and it comes from water. So the oxygen you breathe in gets mostly converted to water as the O2, not CO2. Okay. So here's our two examples one more time. So the fatty acid on top, our glucose on the bottom, all the oxygens are highlighted for you to give you an idea of where every carbon is on the stage. So which of these two molecules has the capacity to generate more ATP, the fatty acid or the glucose?
The acid.
Fatty acid by far, right? Nearly every carbon on average has a higher potential than every other carbon that was on glucose, right? So it's not a fair comparison though. The fatty acid has way more than 6 carbons. And glucose only has 6. This is not a normalized comparison. Let's be fair. So let's say which one has the capacity to generate more ATP per carbon. We're dividing it by the number of carbons to normalize it. What's the answer now?
Still the fatty acid?
It's still the fatty acid, you're right. Because it's still, on average, it was more like the methanol stage than the glucose, which was almost all at the methane stage, where glucose was almost all at the methanol stage. Right? So on average, the carbon is more reduced than it is in glucose. So again, even per carbon, the fatty acid has higher potential. So why do we even bother eating glucose? Let's just eat fatty acids. They taste great, right? Give us way more energy per carbon. Why do we bother with glucose at all?
For us to, as like in terms of crop, right?
I'm sorry, I couldn't hear you.
Is it easier for us to metabolize? Like it takes less effort for your body?
Right. So if you look back at our chart here, and I think that's what you said, the sugar is at the methanol and formaldehyde stage, right? There's already an oxygen on it. So to turn a primary alcohol into an aldehyde, into a carboxylic acid, into CO2, these are all easy redox steps. But turning methane into methanol is not an easy redox step. Right? So that's a difficult step, and that's basically where our fatty acid is. So the fatty acid has a much higher potential to store energy, yes, but it's not easily and rapidly accessible. Whereas the carbohydrate might store less, but it's very rapidly accessible. So if you get a mixture of these two, you have short-term and long-term fuels. That's why we need to get both of them. Okay. Here's an example of one of those coupled reactions. So we're going to take glyceraldehyde 3-phosphate at the top. So this is one of your sugars that you learned in our previous lectures. So it's the simplest aldehyde sugar with 3 carbons, right? And on the 3rd carbon, I've put a phosphate, right? So it's glyceraldehyde, right? We know it's D-glyceraldehyde. We're not going to be concerned with that for this lecture. Everything's going to be a D sugar from here on out. All right, so glyceraldehyde with a phosphate on number 3. We're going to oxidize it. The most easily oxidized piece here is the aldehyde, of course, and it becomes a carboxylic acid. Now we have 3-phosphoglyceric acid. Okay, so this oxidation is not happening in a vacuum. When I oxidize a molecule, I extract electrons from the molecule. Right? So I extracted some electrons from this molecule, and I had to give them to someone. Right? So I give them to another molecule, which was reduced in the process. Okay? So here, I take my glyceraldehyde 3-phosphate, and I extract the electrons, right, and a proton along with it, and give it to NAD+, which is going to be my oxidizing agent, and in itself gets reduced to NADH. Right? And that's a very favorable thing to do. At the same time, we're going to take this molecule, not only oxidize it to a carboxylic acid, but phosphorylate it. I'm going to put a phosphate on. Now, do you think putting the phosphate on this thing is favorable? And if you think it is or is not, give me your reasons.
Is it not favorable because you form the beta keto group?
So if I put the phosphate on here, you're right, there'll be a single bond there, a single bond to the phosphorus, and then a double bond. It will be beta keto. Absolutely right. Any other reason why it would not be favorable? There's one more. That's the best one. You're right. There's one more. Does my 3-phosphoglyceric acid have a charge?
No.
Look at the phosphate. Yeah, it's got an overall -2 charge. The phosphate I'm trying to put on has a negative charge. And when I'm done, I've put 2 negative charges on the same molecule. Right? So I'll put them closer to each other than they were before. So I'm taking like charges and fixing them closer together. That's not favorable. It's not quite the unfavorable thing is making that beta keto arrangement, that's true, but it is still unfavorable. So in order to put this phosphate on here to make this thing, I need to extract some energy in a favorable manner, and that's going to be my oxidation, because oxidation is favorable. So the way this works is I'm going to take the molecule and oxidize it, and at the same time put the phosphate on. I don't do one and then the other. And we'll talk about this next time during glycolysis, because this is one of our reactions in glycolysis. I'm going to oxidize it and put the phosphate on at the same time. Use one to drive the other. And then in the very next step, since I've created this beta keto negative charge next to each other situation, that's my 1,3-bisphosphoglycerate here. Bis is just another way of saying di, right? So 1,3-bisphosphoglycerate. That was one of my 3 molecules in our list, along with creatine and the other one we haven't seen yet. We'll talk about it again on next time. That has more energy than ATP. So I can use it to transfer this phosphate to ADP to make an ATP. This is called substrate level phosphorylation, where I'm going to use the molecule, the substrate itself, as the donor of the phosphate to donate the one phosphate to ADP to make an ATP. Right? Because this molecule, if you go back to our list, was one of 3 molecules that had more energy than ATP did. Okay? So we'll see a couple more like that later. I just want to give you a couple examples with the creatine and this one. We'll see this one again with glycolysis. So in order to do this, there's a lot of redox reaction going on here. We'll see one of those in glycolysis. We need some carriers. Right? We need electron carriers like NAD+ and its cousin FAD. Right? We also have a molecule for biosynthesis, not necessarily catabolic metabolism, but anabolic metabolism. That'll be NADPH. Right? It's got an extra phosphate on it. And for prokaryotes, that's not really a distinction. They just use NAD+ all the time for both. But in eukaryotes, we make the distinction. NAD+ is for catabolic reactions in general, and NADP is going to be for anabolic reactions, not 100% true, but for the vast majority of them that's true. I'll point out the one that is not. And then we also need to carry some carbons around, right? So I need a molecule that can carry carbons, and we'll see a few examples. We already talked about CoA, coenzyme A, and there's two others called lipoamide and thiamine pyrophosphate, or TPP. They're also going to carry carbons around for me, right? And we'll get to those when we get to our pyruvate dehydrogenase complex. Okay. So let's look at some of those molecules. Here's NAD plus on the right, and if you had to draw this, it wouldn't be that difficult because we're going to draw it in pieces. Let's start at the bottom. Isn't this one of our sugars and a nitrogenous base attached to it in black? Which one is that? I have a sugar and one of our nitrogenous bases attached. It has no oxygens.
Honey.
Say again? I couldn't make out what you had said, but this is ribose, and then this is a adenine attached to it, right? So these two together make adenosine, right? And attached to the other side on number 5' is a phosphate. So if you look at just half this molecule, it's AMP, right? Adenosine monophosphate. But there's another phosphate, and this one's attached to a ribose as well. So it looks like a copy of the one below, except instead of an adenine ring over here, we have this unusual ring. It's not a purine, it's not a pyrimidine, it's this heterocyclic ring with a nitrogen in it. And then this amide group on the end, this is nicotinamide. Right, so nicotinamide made from niacin, which we'll see is one of our vitamins. Right, so nicotinamide attached to ribose, attached to a phosphorus, and then adenine attached to a ribose, attached to a phosphate, connected together make a dinucleotide. Right, they're bridged by two phosphates instead of one. It's kind of unusual, but it's a dinucleotide. It's two nucleotide-looking things. Right, that's why it's called nicotinamide, adenine, dinucleotide. And the business end of the molecule is the nicotinamide ring, right? The nitrogen heterocyclic rings where all the business takes place. This is an aromatic ring, and they can rearrange and take on 2 more electrons to become this form. Okay, so it has this form over here, which is missing a double bond, right? And then we rearrange to form the NADH, and you notice it's gained 2 electrons and 1 proton. Right, so these bonds have moved around in here to give you this format. Okay, this one is missing a double bond right here in the picture. Right, and I have 1 extra proton because I only have room for 1 on the ring. So I have NADH, no longer charged. This should say NAD plus down here. And I get an extra proton on the side. FAD is almost the exact same thing. So here's FAD at the bottom. It looks almost identical. I have an adenine ring on a ribose and a phosphate. Here I have a ribose in its straight chain form in blue with a phosphate on its 5' end and a different kind of ring structure. It's not a adenine and it's not a nicotinamide. It's called a flavin ring. It's 3 fused rings. It's not cholesterol at all, right? Got lots of nitrogens and oxygens in here. But this is a flavin ring. And in this case, there are 2 sites, 2 nitrogens that could gain protons and electrons. So when this one gains its electrons, it goes from having 2 nitrogens that are double bonded, conjugated like this, very stable, to each gaining an H and an electron. Right? A double bond migrates to the center. So FAD becomes FADH2. Don't think of it as gaining 2 H's. That's not really what we're concerned with. They're just along for the ride. The important part is it gained 2 electrons. Okay, same thing with NAD. The important part was it gained 2 electrons, right? And the protons again are along for the ride. One gets on the molecule, one rides shotgun. Okay, so NAD and FAD carry my electrons for me. Right, so the other molecule we talked about was NADP, and that's used to carry electrons again, just like NAD was. The only difference is this very bottom phosphate on the 2'. It's the only difference in the 2 molecules. They behave identically in the nicotinamide ring. It again carries 2 electrons. It again carries 1 proton. And we have a phosphate on the 2' of the ribose. It's the only difference. But enzymes can recognize that difference so they know to use NAD versus NADP. Okay. Here's the other carriers we have. So CoA, the one that you don't have to memorize how to draw, but I do want you to know some things about it. If you notice, it has a theme going on here. It's made of an adenine ring in green, a ribose in green, and a pair of phosphates again. This is getting redundant. It's kind of the same architecture over and over. There's a phosphate on the 3' this time. Right, so this part is a building block we keep reusing for all this. Seems important. We have a pantothenic acid unit here, or a pantothenate, right, which you don't have to memorize its structure at all. And at the very end of that, we have a 2-carbon unit, right, 2-carbon group here with a sulfur on one end and a nitrogen on the other. This is an ethylamine, right, so 2 carbons, ethyl, amine, nitrogen, with what we used to call a mercapto unit on it. Mercapto is the archaic name for sulfur, so you could call it a thio or sulfhydryl today. But mercapto is the old-fashioned way of saying sulfur, and the name comes from the fact that if you used to use the old mercury thermometers and you would spill mercury on the floor, you would go grab the powdered yellow sulfur out of the cabinet and throw the sulfur on it, and that would bind to the mercury. So the sulfur was known to capture mercury very well. So sulfur is referred to as mercapto units. And of course we call them thio units now. Right? But the idea here, the business end of this molecule is the sulfur on the end. You may see this written as coenzyme A, all written out, maybe just written as CoA, and sometimes it'll be written as CoA with the sulfur showing. So showing you that's the business part of the molecule where all the reaction takes place. And everything else is abbreviated by the CoA part. But you notice when this binds to a carbon chain, like a fatty acid, right, it attaches through the sulfur to make a thioester bond. Right? Thioester bonds are much like ester bonds, except its oxygen has been replaced with a sulfur. But it has some properties that esters don't have, and it's lacking some properties that esters do. This is shown on the right. You notice a regular ester bond has a resonance structure where the double bond could migrate between the carbon and the other oxygen. Right? This is a less stable arrangement because we have a separation of charges, but it does exist. And it gives this ester bond a double bond characteristic, and again, just like with our peptide bond, it doesn't rotate. Right? But with our thioester, the formation of the double bond between the carbon and the sulfur is extremely unfavorable. Right? So it's really not considered a resonance form. So it only exists in the left form, which restricts its number of resonance structures, therefore it's not as stable. Remember, the more you can draw, the more stable it is. The more stable ones you can draw, the more stable it is. So we make a thioester linkage to these carbon chains, and that's on purpose. We don't want them permanently bound to this thing. It's just a temporary carrier. Right? So I want to carry it on this CoA molecule, which can be dragged along by proteins and other things. And the acyl group, or in the case of 2 carbons, called acetyl group, acyl is the generic name for any number of carbons, gets carried along with it. So this is my way to carry carbons around. Okay? So looking at some things here that carry things for me, I have lots of molecules. We've talked about ATP already. It carries phosphates around for me. The NADs and FADs of the world carry my electrons for me. We have one called FMN, which is very much like FAD. It just doesn't have the other part. Right? And it's also carrying electrons for me. And those are made from your vitamins like niacin and riboflavin. And coenzyme A, we talked about just on the previous slide, has that pantothenate group that can carry a carbon chain for me, an acyl group. Lipoamide can also do that. TPP, we'll talk about, can also carry carbons for me in the aldehyde form. Biotin is another vitamin that can carry carbons for me, but it carries them on the 1 carbon scale, and they must be in the form of CO2. On the other end of the scale, tetrahydrofolate can carry 1 carbon units for me as well, but it generally carries them in the methanol stage, right, or methane stage. Another carrier is S-adenosyl methionine, or SAM, and it again carries 1 carbon units for me, this one in the methyl stage. And then we also can carry glucose on other things like UDPs, phosphates on things like CDPs, and of course nucleotides are carried by nucleoside triphosphates. So these are all the fundamental building blocks of things to build things, and they're also the products of degradation. And they also need to be carried by some molecule. You can't have them just floating around. Most of these carriers are in fact vitamins, or they're made from a vitamin precursor. Here's a list of your B vitamins and some pictures of them at the bottom. And if you look at their list of things, you don't have to memorize this list at all. But if you look at them, they're all involved in these reactions. They all basically become coenzymes for some enzyme in metabolism, right? And the same thing can be said for vitamin C. It's not in this list, but vitamin C is also in that list. It's an antioxidant, it's one of its roles, but it's also a cofactor for hydroxylation reactions. The other ones on this page are your non-coenzyme vitamins. C should not be in this list. But the other ones are the non-coenzyme vitamins, such as the lipid-soluble vitamins, A, D, E, and K. Right? Those are the ones that are involved in various features like blood coagulation, antioxidants, vision, and stuff like that. But these are the fat-soluble vitamins, A, D, E, and K. And if you look at their structures, it should be fairly obvious that they're fat-soluble, not water-soluble. However, vitamin C doesn't belong in this list because it is a cofactor for some enzymes. In fact, it's a cofactor for proline hydroxylase, the one that makes the hydroxyl groups on those prolines that prevents scurvy. But it is an antioxidant. That's probably why they put it in this list, but it has other functions, and that's why I would not put it in this list, because it is a cofactor or coenzyme.